Hi everyone, welcome to part 2 of chapter 2. We are discussing chemical components of the cell. In part 1, we explored what an atom actually entailed with the electrons, the protons, and the neutrons. And then we spent quite a bit of time taking a look at all the different bonds electrons will make between one atom to another to make a larger molecule. So we talked about the difference between a covalent bond, a polar covalent bond, a nonpolar covalent bond, an ionic bond, and a hydrogen bond. And what we'll do in part 2 is we're going to talk a little bit more about bonds, primarily with its interaction when it comes to the pH scale. So what determines what's an acid versus a base versus a neutral environment. And then the remainder of the chapter, or I should say the remainder of part 2, is going to take a look at macromolecules such as carbs, proteins, lipids, and nucleic acids. And we'll take a look at what they're actually built out of and how the covalent and the non-covalent bonds play a role in their overall structure. So without further ado, let's go ahead and start exploring part 2 of chapter 2. All right, now what we see happening towards the end of our chemical bond discussion is that there are some polar molecules, primarily water, that are essential for performing or determining the pH of a solution. And this all has to do with the fact that we can grab any solution and we can basically take a look at, when it's exposed to the right environment, what its ratio of hydrogen versus hydroxide release is going to be. So when you do pH, you can discuss it many different ways. And the book goes about it in discussing the fact that there are polar molecules that once they interact with water will form either acids or bases, and the fact that the proton from one water molecule will often migrate over to the solution and help it dissolve into different components. But at the core of it, just to kind of say it straightforward, what determines the pH of a solution is always going to be the amount of hydrogen versus hydroxide ions that a solution has. So when we mix it with water and it dissociates, it falls apart, how many hydrogens versus how many hydroxide ions does it have? And we basically have three scenarios, right? You can have scenario number 1 where the hydrogen exceeds the hydroxide. Scenario number 2 will be that the hydrogen equals the hydroxide. And then scenario number 3 will be that the hydroxide exceeds the hydrogen. So those are your three options if you're going to do a ratio between two items. So hydrogen exceeds hydroxide, hydrogen equals hydroxide, and hydrogen is less than hydroxide, or hydroxide exceeds hydrogen, right? So you can take any solution that you want and you can basically pull it apart, and from a chemical standpoint, you would literally quantify how many hydrogens, how many H's versus hydroxide, OH's, this solution has. And if you see that the hydrogen exceeds the hydroxide, such as our first option right here, then we are looking at an acid. If we see that it's equal, then this would then be considered a neutral solution. And our last option, if the hydroxide exceeds the hydrogen, then you're looking at a base. And instead of base, some people like to say alkaline solution. Both answers are completely acceptable. Now, it's very easy to quantify pH, because it turns out that there is a pH scale out there that will run from 0 to 14, and the way it's going to work is that 7 in the middle will be our neutral territory, and anything below 7 is going to be considered an acid, anything above 7 is going to be considered a base. So let's go ahead and take a look at the next slide, and let's see exactly how that scale is going to be running. All right, so here we have our pH scale. As you can see right here in the second column, the pH scale from a quantitative standpoint will run from 0 to 14. And what you can also see in the first column is that it's taking a look at the amount of hydrogen ions that a solution has, okay? And what we're going to find out is that as the scale goes from 0 to 14, the amount of hydrogen starts to decrease. And it's all about the fact that we are trying to use the relationship between the hydrogen versus the hydroxide to explain a pH of the solution. So let's start with the easy one. Let's start with the number 7. That's right in the middle of the solution. Oh, my apologies. I completely forgot to show you. That's right over here in the third column is the concentration of hydroxide ions. And what we're going to see is that they're going to start off really low. See how it has a negative number? And as it goes down, the number will slowly start to increase. Okay, so what we see happening is that on top of our table, we have a lot of hydrogens and very low amount of hydroxide. And at the bottom of the table, we see that our hydrogen, you can see it's in the negative 14 power, is very low, where the hydroxide has reached its max when it comes to this table. There you go. And then now if you take a look at 7, you're going to notice that both numbers are the same. So if hydrogen equals hydroxide, we are looking at a neutral solution. So a pH of 7 right in the middle is considered a neutral solution because the amount of hydrogen equals that of hydroxide. And probably the best example of a neutral solution is pure water, good old-fashioned H2O. And in fact, if you take a look at H2O and you were to pull it apart, you would have one hydrogen and you would have one hydroxide. And that explains why it's a neutral solution. There's also other examples of neutral solutions. For instance, when it comes to humans, our blood and our cerebral spinal fluid is actually pretty close to neutral. It's around 7.3. So there are lots of different examples out there of a neutral solution, but at your core, what you want to keep in mind is that when you hear neutral solution, 1, the pH is 7, and 2, the concentration of hydrogen equals that of hydroxide. Now, once we've established the neutrality, so we're at 7, what we're going to see happening if we go back to the pH scale is that any number below 7 is going to be considered an acid. And the way it's going to work is the lower the number gets, the more acidic the solution becomes. And the way you're going to be able to explain that is because if you take a look at your hydrogen concentration from 6 and you work your way up, what you're going to notice is that the amount of hydrogens in the solution starts to increase the lower the number gets, okay? And the lower the number gets and the more hydrogens you accumulate, the more acidic the environment becomes. So any number below 7 is considered an acid. The lower the number, the more acidic, and that's because you have an increase in hydrogen concentration as you start to decrease the pH number, allowing for a stronger acidic environment to form. Now, because we're doing this in logs of 10, it means that each difference in the number when it comes to the pH scale, so if you go from a 1 to a 2 to a 3 to a 4, is a multiple of 10. So there's a tenfold increase if you're doing a difference in the acidic environment. What do I mean with that? Well, let's say I say to you on an exam, I say to you, you're looking at 2 solutions. One is a pH of 2, and the other one is a pH of 4, right? So I can ask you which one is the most acidic, and hopefully we'll go, all right, the lower number is the most acidic, so I'm going to pick pH of 2. But I can also say to you, out of both solutions, which one has the most hydrogen ions, right? And we're going to pick 2 again, because we know that as the numbers start to decrease in the pH scale, the number of hydrogen goes up, and that's what makes it more acidic. How about if I say to you, between 2 and 4, which one has the most hydroxides? Which one has the most hydroxide ions? So OH. Well, hopefully in that case, we go with a pH of 4, because the ratio between hydrogen and hydroxide It's different in 4 than it is in 2, and we know in 2 it has more hydrogens, which means that the hydroxide starts to go down, right? So if I say to you which one has more hydroxide, that would be a 4. And you can double check that on the scale. You'll notice that when you look at your hydroxide concentration, the numbers start going up as you progress from a low pH over towards the neutral territory of 7. Okay, and it's all about that ratio. So if the hydrogen is high, that means that the hydroxide is low, or I should say, if the hydrogen is increasing, that means that your hydroxide is decreasing. I can also say to you, okay, so you have a pH of 2 and you have a pH of 4. How much more acidic is the 2 compared to the 4? Right? So how much more acidic is a pH of 2 when you compare it to 4? And the way you're going to answer that question is you're basically going to take a look at how many steps does it take on the scale to go from 2 to 4. And each step is going to be a log of 10 or a multiple of 10. So if I start off at 2, my first step is going to bring me to 3. That's going to be a 10-fold increase. And if I now go from 3 to 4, there's another 10-fold increase. So that means that if I have to go from 2 to 4, I'm going to have to do 2 steps, which means it's going to be 10 times 10 is going to be 100 times more acidic. If you had to do 3 steps, so maybe you were going to go from 2 to 5, it would be 10 times 10 times 10 would be 1,000-fold change. If you add one more step to that, now we're looking at 10,000. Another one, 100,000. And you just keep going. Each step is a log of 10. So there's a 10-fold change between your numbers on your pH scale. So thinking about that, now your scale from 0 to 14 shows you a huge range of characteristics from the top, the 0, all the way to the 14. Because each step that you do is going to be a 10-fold increase. All right, so one more time. The pH of 7 is neutral. Hydrogen equals hydroxide. Any number below 7 is going to be considered an acid. The lower the number, so let me write it down over here. So any number below 7 is considered an acid. The lower the number, the more acidic. And that is because the lower the number, the more hydrogens you obtain, the less hydroxides you have in that solution. And this amount of dissociation and the change of ratio is then how you explain the difference between a pH of 6 versus a pH of 0 when it comes to acids. So if someone were to ask you, why is the 0 more acidic than the pH of 6? You would say, well, the 0 has a higher amount of hydrogens versus hydroxides when you dissociate the liquid or whatever solution you're looking for. Go ahead and take a look at the last column. You're going to notice some solutions and their pH values. So we have battery acids at 0.5. We have our stomach acid, which accounts for why our stomach is the most acidic part of our body, because its acids can range anywhere from 1.5 to 3.5. We see some of our popular drinks, lemon juice, cola, orange juice. Most of the drinks that we consume are actually considered acidic. Oh, here's our beer. We can't forget about the beer. How about coffee? Any coffee lovers in class? Oh, I love me a good cup of coffee. Look at this. Black coffee is a pH of 5, and an acid rain is 5.6. Oh my goodness. Why would they put the two of those together? Some of us might never drink coffee again. Now, don't stress yourself out that a lot of the drinks that we consume are acidic. Our body has buffers that will protect us against overall pH changes, and one of the largest buffers that we actually utilize is our saliva and the mucus. So when you expose your digestive system to a soda or to orange juice or even to coffee and beer, we don't feel the difference in their pH, because our saliva and our mucus will quickly neutralize the acidic component so that it doesn't actually upset our digestive tract as it moves from, our oral cavity down into our stomach and into our small intestines. And a really good example of our buffers at work or when they're not working is whenever you have a cold or you're not feeling as well and you're kind of stuffed up, you're going to notice that your mucus, your buffer isn't working as properly. So if you go and you drink yourself some orange juice, it's going to kind of burn your esophagus as it moves down your digestive tract. And that's because your buffers aren't functioning at optimal capacity because you're not feeling well. All right, there's no need to memorize the solutions and their pH value. On your exam, I'm going to give you your own set of solutions and I will be providing their pH. And then what I'll ask you is if it's an acid, a neutral, which one has more hydrogen, which one has more hydroxide, how much more acidic, and all that other good stuff. Now, let's take a look at the bottom part of the chart, and that is going to be your basic or your alkaline solution. Now, the way that one's going to work is that the higher the number, the more basic the solution is. So anything above 7 is considered a basic. The higher the number, so I should go from 7 to 8 to 9 to 10, the more basic or alkaline it becomes. And that is corresponded by the fact that as the numbers start to increase, we see that our hydroxide ratio starts to increase. And if the hydroxide is going up, that means that if I come over here and take a look at my hydrogen, I'm going to notice that it's starting to go down. So very similar to what we saw above the 7, when you now come below the numbers, we're going to see that any number that's higher than a 7 is going to be a base. The higher the number, the more basic or alkaline, and that's all because, once again, you're looking at your ratio, and the higher the number becomes, the more hydroxide ions you gather, the less hydrogen ions the solution has. And just like in the acids, each step that you take will be a tenfold increase when you take a look at your alkaline solutions. So let's take a look at some of the examples. We have seawater at 8. We have hand soap. A lot of our cleaning solutions are very basic. 9.5. Bleach is there at 12.5. Caustic soda at 13.5, and we have our detergent and our ammonia in the 11.9 to 12 ratio. So go ahead and make a little notation for yourself to come back to this chart because you want to definitely be familiar with it because I do tend to ask a handful of questions on exams whenever we covered pHs because I really want you to understand the whole fact that it's a ratio between hydrogen and hydroxide and of course how the numbers correlate to a solution being an acid, a neutral, or a basic/alkaline solution. On to our last topic of this particular chapter. Actually, my apologies, the second to last topic of this chapter. We're going to take a look at some of the small molecules in the cell. And what we see happening is that overall in the human body, if we disregard water molecules, it turns out that all the molecules in our body are basically composed of at least some percentage of carbon. And that is then in essence why we like to go ahead and we like to classify molecules based on if they're organic or they're inorganic. So the way it's going to work is that if a compound contains carbon, it's going to be classified as organic. If you don't see any mention of carbon, then that compound is considered inorganic. So water, believe it or not, H2O, has absolutely no carbon inside of it, so it is technically classified as an inorganic molecule. Whereas if you look at your carbohydrates, it's full of carbon, so that would be an example of an organic molecule. One of the reasons why carbon is so popular is because of the fact that it is able to generate 4 different bonds, and it turns out that whenever you match a carbon to a carbon, it actually forms a very stable structure, and that structure can then be used as a backbone to provide support for the for the compound that you're forming. So we'll see it in a lot of items as we go ahead and we start looking at our macromolecules. So carbon is a very popular choice. And in case you're wondering, wait, we had carbon already with the most commonly found elements in the human body. What are the other three? Hydrogen, oxygen, and nitrogen. You're completely correct. All right, so having said that, let's go ahead and take a look at some of the other contents within the small molecules in the cell section. Let's go ahead and take a look at that cells contain 4 major families of small organic molecules, and from there we'll take a look at the sugars, the fatty acids, the amino acids, and the nucleotides. All right, so all together within the human body, we basically can go ahead and when we take a look at our organic molecules, so those that contain carbon, we basically see that there are 4 macromers or polymers, I should say, large organic molecules that you'll find in pretty much every single cell. So they're right over here indicated in yellow. This is going to be your polymer or your macromolecule. Poly means many, so it just indicates that it's a larger structure. You're going to have your polysaccharides, your glycogen, and your starch. These will collectively fall within the carbs. So these are your carbohydrates. Then you're going to have your fats and your membrane lipids. So these are just kind of like your lipids, which are your fats, and your proteins and your nucleic acids. And what you can do is you can go over here to the gray section and we can see what the monomers or the building blocks are of these polymers, these large molecules. So the carbs are composed of simple sugars. Your fats are composed of fatty acids, also a little bit of glycerol, but fatty acids is what the book likes to focus on. Proteins are composed of amino acids, and your nucleic acids are going to be composed of your nucleotides. Now, what we'll do over the next few slides is we'll take a look at each of these categories, and we'll kind of explore the relationship between the polymer and the monomer a little bit more. So let's start off by taking a look at our sugars, at the carbs. Okay, so sugars, carbs, are considered both an energy source and they're subunits of polysaccharides. Now, the way it's going to work is that your body likes to build. It likes to start off small with a very simplistic sugar, and we like to call that a monosaccharide. Saccharide means sugar, mono means one. And probably the best known monosaccharide is what we consider our blood sugar, the sugar that our body loves to utilize for cellular respiration, and that's good old-fashioned glucose. Now, a monosaccharide can be combined to create a disaccharide, meaning that you have 2 sugars attached, and then you can keep building. And you can get a polysaccharide, which means many sugars, an oligosaccharide, and then eventually you get to the highest level, which is that you start looking at your carbs. And it turns out that within the carbs, you can have many different functions. So you can have carbs, for instance, like starch, or glycogen. These are excellent energy sources. So for instance, whenever you have a meal and you have a starchy food like a potato or a pasta, your digestive system will quickly take up these carbs and break them down and release all those millions of sugars that you find within the cells, aka glucose, so it can go ahead and fuel the body in the form of cellular respiration, that the mitochondria is able to convert the energy from the glucose into ATP molecules. We can also use some of the carbs for structural components, like for instance, another carb that's well studied is called cellulose. Cellulose you heard mentioned in your previous chapter because it makes up the cell wall of plants. How about chitin or chitin, as some of you like to say? Chitin is found in the exoskeleton of a lot of insects as well as in the cell wall of fungi. So it plays a very important structural role within these cells. In fact, if you've ever stepped on like a little a spider or like a little bug, like maybe you saw a little cockroach and you stepped on it and you hear that cracking sound, that's the chitin molecules that are contributing to that particular formation. Now, one thing you want to keep in mind as we're doing this particular discussion is you want to be able to explain exactly how do you build from a monomer to a polymer, and how exactly do you destroy or break down from a polymer to the monomer. And it turns out that regardless of what class of molecules you're looking at, whether you're looking at your sugars, your proteins, or your fats, you're always going to see two of the same reactions occurring. And in order to talk about those two reactions, we're going to need to flip over to the next slide so we can explore it a little bit more. So here we go. The two reactions that you want to keep in mind are condensation and hydrolysis. And it talks about the fact that when you look at your macromolecules, and your monomers, we see that regardless if you're building or you're destroying and pulling apart, you will always have to evolve a good old-fashioned water molecule. So let me try to explain this for you based on the illustration. Let's start on top of the image. And what we see happening here is that they went ahead and they drew two monosaccharides for you. So these are two individual sugars. And what we see happening is if we want to take those two monosaccharides and we want to build, so we want to combine them, we're going to follow this arrow on their left-hand side. We want to make a disaccharide, something that's a little bit larger. In order to do that, we're going to need to go ahead and empty out a space on the chemical structure of both of those monosaccharides so that they can bind together. And in order to create a space, we're going to go over to one monosaccharide and remove a hydroxy group so that we create a little empty space on that bond. And then we're going to go over to the second monosaccharide and we're going to remove a hydrogen ion. Once again, our goal is that if we take away these elements, we create an empty space so that the two monosaccharides can link together. Now that the hydroxide and the hydrogen have been removed, there's an empty spot on both of the monosaccharides so that they can successfully link together. And when they link together, they create a disaccharide. So you've built something that's larger. Now, what do we do with our waste products, right? Because we have an OH that we removed from one side and a hydrogen that we removed from the other side. Well, it turns out that if you combine these two, you get water. And water is considered a waste product in this particular example because you no longer need it. So we're going to expel it. We're going to get rid of it. And when you expel it, we then call it a condensation reaction. Now, some of you might have heard of condensation by a different name, and that's because it also goes by dehydration experiment. A dehydration synthesis basically means that we're doing a condensation reaction. Both names are acceptable, and it indicates the fact that we are building, so we're getting from monomer to polymer, and as we're doing so, we're losing water along the way. The water is expelled, no longer needed. It's considered a waste product. All right, so here it says, in a condensation reaction, aka a dehydration synthesis, the two molecules need to join together, and as a result, they will lose a water molecule. Okay, so condensation, we're building, and we're losing water as a result. Now, the other option is going to be the opposite. So let me just kind of clean this up a little bit so we don't get overly confused. All right, so now let's start at the bottom of the slide. And what we see happening here is that we have, and I'm going to switch colors too, we have a disaccharide, and our goal is to lyse it or pull it apart so that we can have two individual monosaccharides. So that sounds easy enough, right? You just kind of find where they're held together. Here's our little glycosidic bond, which is a version of a covalent bond. And what we're going to do is we're just going to destroy this bond. So you take it and you pull it apart, you lyse it so that the two individual ones are once again by themselves. Now, when you do that, it turns out that there's an empty spot on both of them, because remember, they were bonded together. So you have to fill up that empty spot. And what you're going to do is you're going to go ahead and restore the hydrogen that was initially removed from one and the hydroxide that was initially removed from the other. We're going to restore it back to its original location. Now, what molecule can you think of that we can utilize that will give us access to a hydrogen and a hydroxide? Good old-fashioned water. That's right. So it turns out that when you are destroying, when you're going from large to small, you're going to have to use water to make sure that the monomers, your end products, are completely stable all by themselves. And in order to do that, we're going to go ahead and do hydrolysis. Lysis indicates that you're pulling it apart. Hydro means that you're using water to get it completely successfully done. And that's because the water molecule will go into the reaction, and as it goes into the reaction, it's pulled apart so that hydroxide can be provided to one and hydrogen can be provided to the other. So you actually consume the water and it's utilized to restore the monosaccharides back to their original condition. A really good example of hydrolysis is anytime you consume a meal, your body, your digestive system will do hydrolysis. It will use water to pull the food apart into their individual building blocks so we can extract as much energy from it as possible. So right over here at the text, it says the reverse is going to be hydrolysis. And that means that we're adding water to the reaction, and the goal is obviously to go from polymer back to monomer. So you're lysing, you are making a smaller component. So condensation is when you're building and you lose water due to the fact that water becomes a waste product and you remove a hydroxide from one and a hydrogen from the other to produce your final product. Hydrolysis, lysis means to pull apart, is when you go from large to small. You're pulling it apart, and in order to be completely successful, you're going to have to use up a water molecule so that you can restore the hydroxide on one side of the monomer and a hydrogen on the other side of the monomer. Now, this is very important, so please take your time to process this information, because the condensation/dehydration and the hydrolysis reaction will be consistently utilized as we're building and we're destroying. So in a little bit, when we start talking about proteins, if you want to have amino acids become proteins, you're going to have to do condensation. If you want to have proteins be broken down into amino acids, you're going to have to be doing hydrolysis. So you're going to see these two names, these two reactions consistently, and they're the opposite of each other. One is to build, to pull together, and the other one is to pull apart and to destroy. Here you go. So here's another illustration showing you how they're basically reverse reactions from each other. Let me switch back to my red color. So you can see that if you have two pieces A and B that you want to put together, you want to create a larger structure, you're going to have to remove the hydrogen on one side, hydroxide on the other, and these then become the water molecule that you're expelling. And this is then your condensation reaction. In order to build, it always requires energy. So that's why over here they went ahead and they wrote down it's energetically unfavorable. That's because your body has to spend energy to get this reaction done. It takes more effort to build than it does to destroy. We're going to have a whole chapter on energy where we'll talk about the consumption and the production of your ATP molecules. Going back to our chart, we can see that now that we have a larger component, if we want to go back to the individual building blocks of just A and B, we're going to go ahead and have to do hydrolysis. And as you're doing hydrolysis, you're pulling the bond apart that was initially holding together your larger complex. And as the bond falls apart, we're going to have to return the hydrogen and the hydroxide to their original location. So to do that, we're going to recruit over a water molecule and we're going to lyse it. We're going to destroy it so that we can go ahead and restore it over to the monomer side. Now, anytime you break a bond and you pull stuff apart, the bond actually is full of energy. So you release that energy into your system. So what we see happening is that hydrolysis is energetically favorable. And this kind of makes sense because when your digestive system pulls apart the food that you're consuming, all that energy that the food has is released through the bonds as they're being broken. So your digestive system can then go ahead and convert that energy into ATP molecules, which we then use to kind of fuel the system throughout the day. All right, so we talked about our carbohydrates. Those are composed of sugars. So now let's take a look at some of the fatty components within the cell. And let's start by looking at our fatty acid chains. We find them in many different ways in the cell, but most of us are probably familiar with them due to the fact that they're a component in the cell membrane. And the reason the fatty acid chain is such an important one is because it has both a hydrophilic and a hydrophobic component. It's hydrophilic, we like to say it has a hydrophilic head, and it has a fatty acid tail that's mostly composed of hydrophobic components. It turns out that the orientation of these fatty acids, how they're layered together, are going to be very important for the selective permeability of a cell membrane, which we're going to talk about in just 1 second. But before we do that, I want to talk about the fact that lipids are considered hydrophobic, meaning that they are water-fearing. They do not like to interact with water by themselves. Lipids have a high amount of energy that they hold on to, very similar to the carbs. They actually have more energy than carbs. However, your body favors to get its energy from carbohydrates over lipids because lipids are hydrophobic. Your body would have to put in more effort to get the water molecule to interact with the lipids to extract the energy out of it, where it has an easier time just getting its maximum energy from the carbs. In order for us to successfully process lipids, we will always need to utilize an emulsifying agent, something that can freely interact the water and the lipids together. And the emulsifying agent that our digestive system uses is good old-fashioned bile. Bile, which is produced by the liver and stored in the gallbladder, is our emulsifying agent. Anytime you have a meal that contains fat, bile is released in the system, and it allows the water molecules in your digestive system to interact with the lipids that you've consumed. Thereby, it can increase its efficiency when it does hydrolysis, and it pulls apart the lipids into its fatty acids and glycerol components. Now, another thing you want to think about with fats is that you can classify them based on if they are saturated versus unsaturated. And all that will have to do with the composition of the fatty acid tail. Now, a saturated fat is going to be a fat that when we look at the fatty acid tail is going to be completely filled with single bonds. Lots and lots of single bonds. And these single bonds are going to allow for the fatty acid tail to be very rigid. And because of that, this fat is going to be a solid fat. And examples of solid fats are going to be our meat and dairy products. So anytime you consume things like bacon, what you're doing is you're consuming a saturated fat. If you guys like to cook and like let's say you buy chicken, a lot of times you'll clean the chicken and you'll find like those adipose, those little fatty pieces that clump together. That's an example of a saturated fat. If you have a nice juicy steak, like you went over to a restaurant, you got yourself a delicious steak, you weren't able to eat all of it so you took it home. The next day when you take it out of the refrigerator, you notice the marble on it, the little white layer that forms over it. That's an example of a saturated fat. So a saturated fat is a solid fat. It is composed of nothing but single bonds within its fatty acid tail, which allows it to become solid. And like I said to you, your meat and your dairy products will fall right into this category. On the other hand, what we see is that some fats are considered unsaturated. And unsaturated fats means that within the fatty acid tail, there will be some double bonds. And these double bonds are really important because it's going to allow for the fatty acid tail to have what we call kinks. Here we go. And kinks is what we see in drawn like here, meaning that it can't lie straight. It will not have a very rigid structure, and because of that, the unsaturated fat will be liquid. Unsaturated fats are plant-based fats. So if you have, for instance, probably the best one is like olive oil. If you have olive oil in your pantry, it will be a liquid fat. We can even just have it out on our countertop. It will never solidify. Plant-based fats are going to be a really good example of an unsaturated fat because they have lots of kinks within their fatty acid tail, allowing them to stay liquid. Now, if you were to do a comparison between saturated fats and unsaturated fats, the fact that saturated fats tend to become solid makes them way less healthier compared to unsaturated fats. In fact, you can go ahead and take a look at the text that's on the right-hand side, and it says, although fats are essential in the diet, saturated fats tend to raise the concentration of cholesterol in the blood, which tends to increase your chances of clogging up your arteries, increasing the risk of heart attacks and strokes. So fatty acids, my apologies, saturated fats are less healthy compared to unsaturated fats. Your body has a slightly easier time breaking apart the liquid unsaturated fats. But keep in mind, at the end of the day, both of these fats are hydrophobic, and in large quantities, they could be not healthy or detrimental to the body. I also want to bring up something else, which is that some of you might have heard of trans fats. So what's a trans fat? Well, a trans fat is a fat that was originally unsaturated, meaning it was liquid, but some of the kinks, some of the double bonds were biochemically removed. So in a lab, they went and they removed some of those double bonds. And the reason they did that is because they wanted the liquid aspect of the unsaturated fats to turn into a solid. Because from a restaurant standpoint, it turns out that it's easier to transport, store, and use solid products than it is liquid products. So the thought process for those biochemists was, let's take the fat that's healthier, or unsaturated liquid fat, and let's just manipulate it a little bit so we can get it in the form that we really want, which is going to be the solid form. And that is then what a trans fat is. So it's a manipulated, unsaturated fat that has been turned into a solid. Well, wouldn't you know that when you turn it into a solid, that unsaturated fat becomes just as unhealthy as a saturated fat. So starting a few years ago, a lot of research went into the fact that we want to remove trans fats from our diets. So a lot of restaurants will advertise the fact that they no longer use trans fats and they've switched over to healthier alternatives. And in fact, some states, like for instance New York, have actually banned the use of trans fats by restaurants. So just a little FYI. Now, going back to the use of the fatty acids in the cell membrane, what we see happening is because they have that hydrophilic component by their phosphate glycerol head, and they have that hydrophobic quality in their fatty acid tail, what we see happening within our cell membrane is that it will take the fatty acid phospholipid molecule, and it will create a bilayer. It will stack two layers on top of each other. Here's the little bilayer. Here's one layer. Here's the next. And when it stacks the bilayer together, it will go ahead and expose the hydrophilic part of it, the hydrophilic head. Remember, hydrophilic means water-loving. It will expose that to the external as well as internal components of the cell, where you find lots and lots of water. And that's okay because the hydrophilic head loves to interact with water. On the other hand, the hydrophobic tail, the water-feeling tail, will be tucked in inside. So it's not exposed to any of the water components, whether it's inside or outside of the cell. Now, one of the main reasons why this is such a brilliant setup is because it turns out that the phospholipid, with its combination of hydrophilic and hydrophobic quality, is one of the main reasons why cell membranes have a selective permeability. They will be able to control what comes in and what comes out. If the membrane was fully hydrophilic, then anything would just float in and out of the cell. If it was fully hydrophobic, then really nothing would be able to flow in and out of the cell, because keep in mind that our body is just inundated with water molecules. So the arrangement of the hydrophilic head and the hydrophobic tail, or I should say the hydrophilic head being exposed to the water component and the hydrophobic tail being tucked away, is a perfect scenario for our plasma membrane to allow for selective permeability to occur. Now, finishing up our macromolecules versus monomers, we can go ahead and we can take a look at amino acids. Amino acids are the monomers of proteins, and when they link together due to a dehydration or condensation reaction, we see that they'll actually form a very specialized covalent bond called a peptide bond. And this peptide bond is very unique. It's only found when amino acids will link together. And the structure of these amino acids, we have about 20 of them, is very predictable. Now, what I mean with that is go ahead and take a look at your illustration. And right over here, we see an example of an amino acid. And all amino acids, when you look at their chemical composition, will have a carbon in the middle. And that carbon will be bound to 4 different items. On one side, it will just have a simple hydrogen, which is perfectly fine. On the other side, it will have an amino group. And that amino group, as you can see, is going to be composed of a nitrogen and 2 hydrogens. Opposite of the amino group will be a carboxyl group. The carboxyl group consists of a carbon, an oxygen, and a hydroxide molecule. Now, the amino group and the carboxyl group will always be opposites from each other. And now what we see happening is that opposite from the hydrogen, we will have what we like to call a side chain, which is often indicated by an R. Now, I said to you that there are 20 amino acids out there, and out of all the 20, if you were to sit there and you were to draw them out, you would always have the carbon in the middle, the amino group, the carboxyl group, and the free-floating hydrogen. What would make the 20 difference is whatever you put in the R group, in the side chain. So here you can see that this is an example where they put a carbon and 3 hydrogens. But you can basically put whatever you want in there, and whatever you put in the side chain will then determine which amino acid you're looking at. So you're looking at glycerol, or you're looking at saline. I can't even think of these amino acids. I'm so sorry. But there's lots of variation within the amino groups itself. And you know what, guys? When we get to chapter 4, we're going to discuss all 20 of these amino acids, and we'll talk more about the amino group versus the carboxyl group. Valine, that's what I was looking for. Sorry. Now, keep in mind that when you're linking together your amino acids, you are still doing your dehydration / condensation reaction to build your protein. And if you have your protein and you're trying to destroy it or pull it apart, you're still going to be doing your hydrolysis reaction. Now, I did make a statement on this PowerPoint, and this one is really important to point out. And like I said, in chapter 4, we're going to be exploring it a little bit more. But it turns out that when you're building a protein, your amino acids are very specific as to where they get their hydrogen and their hydroxide from. And it turns out that in order to make a successful peptide bond, you will have to go over to the amino group. So right over here, go over to the amino group, and that is where you're going to remove the hydrogen. And then you're going to go over to the carboxyl group of the other amino acid. Your carboxyl group is this area right over here. And that is where you're going to be removing your hydroxide ion. So if you're doing your condensation reaction, then the hydrogen from the amino acids from the hydrogen from the amino group from one of the amino acids will have to interact with the hydroxide from the carboxyl group from the second amino acid, and that is then how you're going to form your H2O molecule, which is discarded or getting rid of whenever you do your building through your condensation or dehydration synthesis. Now, we're gonna talk more about proteins over and over again. So for now, I'm just gonna leave it here, the fact that they're composed of amino acids, that they have a peptide bond, and that they're very specific when it comes to removing their hydrogen from the amino group and the hydroxide from the carboxyl group. We're going to discuss that more in chapter 4. I just kind of want to look at our last macromolecule, which are going to be our nucleic acids. These are going to be things like your DNA molecule, your RNA molecule, and what we see happening is that their monomer, their subunits are going to be the nucleotides. So we talked about this in chapter 1 already. The nucleotides are the letters, so your adenine, your thymine, your guanine, your cytosine, and in the case of RNA, there's no thymine, so instead we have uracil. So those are going to be the building blocks that will give us our DNA as well as our RNA, because those are all examples of nucleic acids. All right, so now that we've tackled our small monomers, how they're building to make macromolecules or polymers, let's just go ahead and take a look at how some of these macromolecules will actually behave within the cell. And what it means is that we basically want to see how a covalent versus a non-covalent bond can be applied to the macromolecules to increase their overall function within the cell. So go ahead and take a look at figure 229, and you can see that macromolecules are quite abundant in the cell. So we can see right here, this is a bacterial cell, but it's very similar for a human cell. We see that it's mostly composed of water, which shouldn't surprise anybody. And then the other 30% is made out of different chemicals. And within the chemicals, you can see that the macromolecules, your carbs, your proteins, your nucleic acids, and your phospholipids really make up the majority of the chemicals that you will find. And now that we've also discussed the many different roles they have, hopefully it makes sense as to why your body wants to utilize them for its day-to-day activity. Now, we also discussed the fact that a lot of the molecules will use covalent bonds and not covalent bonds to kind of build upon the monomer to the polymer situation. And obviously those bonds also need to be destroyed whenever you're doing your hydrolysis. But what we also need to talk about is the fact that oftentimes the macromolecules will have to interact with each other, and when they do so, they can utilize different non-covalent bonds to kind of figure out if they want to have a proper reaction, if they want to have a weak bond, or if they want to utilize it more of a building mechanism. So on figure 2.35, it shows you that you have a macromolecule A that will go ahead and encounter other macromolecules that are labeled B, C, and D. Now, why do we care about this? Well, we know that our body is 30% made of different chemical reactions, so the macromolecules will interact with each other. And in order for them to fully interact, we'll see that they'll utilize lots of non-covalent types of bonds to see if they want to have more of a temporary or a permanent type of reaction. So you can see in the middle right over here, that with all of the interactions, A with B, A with C, A with D, you have these little red lines that will form. And these represent our non-covalent bonds. And what we see happening is that when it comes to A interacting with B and A interacting with C, it's really not a good match. They're not able to form a successful bond, which means that it takes very little effort to kind of allow them to kind of break apart. So they're not a match, meaning that they really must not need to interact with each other for the body to be successful. On the other hand, when it comes to A interacting with D, we see that the non-covalent bonds are not only more numerous, they also tend to stick together and they're not as easily broken apart. So that will then indicate two macromolecules that can freely interact with each other, and hopefully they'll be able to produce a more successful result together versus apart. Now, I also want to point out to you that the book comes with different animations. So as you can see right here on the notation for figure 2.35, there's a little movie that corresponds with the information. These animations have actually been linked within D2L itself, and you can also go over to the student companion site to take a look at it. So I highly encourage you to go ahead and take a look at some of the animations. Like I said to you, there is a specific folder on the side of D2L that's labeled animation, so you can just click through that, or you can go through the ebook/student companion site, and you'll also find a list of the animations there as well. One more slide to go, so let's go ahead and see what that one's all about. All right, now this one talks about the fact that it's quite common to have both a combination of covalent as well as non-covalent bonds that will cooperate together in order to to reach the final structure of a macromolecule. Now in this case, we're looking at a ribosome, and a ribosome is composed of two components. It has a large subunit and a small subunit that have to click together in order for it to perform at maximum levels. So if we start all the way on the left-hand side of the image, we see that we have our subunits, our monomers. Those are our amino acids and our nucleotides, and we see that they will go ahead and they'll link together through a covalent bond as they're doing their condensation reaction to form their macromolecules. So you can go ahead and you can see protein is being formed right over here because the amino acids have covalently bonded together through their peptide bond. And then we can see our RNA molecule has formed here by combining together the nucleotides in a covalent bond. Now, the non-covalent bond in this case will be utilized to go ahead and combine the two polymers and allow them to get to the final functioning form of our product, which in this case is going to be our macromolecule of a ribosome. So the non-covalent bonds in this example are being utilized for structural support and to get to our final end product. Alrighty, having said that, this is a nice place to kind of end chapter 2. As always, I want to remind you that I am available through email and my virtual office hours. So please don't be shy. Let me know if you have any questions, comments, or concerns. And of course, feel free to use the discussion board as well. Now we are going to skip chapter 3 for now. We're going to encounter it for a next exam, and we're going to move on to chapter 4, which is going to give us a much more detailed look on proteins. We're going to become protein experts, right? All right, so whenever you guys are ready and you're done processing chapter 2, go ahead and let's talk about chapter 4, which will be about the proteins. All right, see you in the next lecture.